What is genomics? Genomics is an interdisciplinary field. It is a field, a steady field in biology, focusing on the structure, function, evolution, mapping, and editing of genomes. It is all about what? Genomes. Its name is genomics. It comes. The name genomics comes from because it is study about genomes of these features. These features of genomes. Metagenomics is study of a collection of genetic material, that is genomes, from a mixed community of organisms. And metagenomics usually refers to the study of microbial communities. Here is very interesting picture about about genomics. Here is genetic paternity test. You know, when there will be argument for the paternity, then there will be testing for that parent. The father's DNA will be taken, and the son or the daughter's DNA will be taken, and then it will be uh, identified by that or confirmed by that. Then that is genetic. Genomics is important here. Genetic compatibility test. Is there genetic compatibility? How much percent? 100%, 90%, or that is genetic compatibility test. Is there genetic fingerprinting? You know, this is a fingerprint printing. That is, every individual is different from the other by his or her fingerprint. This is because of the DNA sequence variation. So that is it. Genetic ancestry test, genetic disease risk. It is possible to identify genetic disease risk by thanks to this field of study, genomics. Personalized medicine. It is possible to formulate individual medicine or personalized medicine based on the person's genetic makeup and the disease type. This is because of this is possible because of genomic science. Very interesting and amazing science, which also I like the most. This is about metagenomics. Here is the process of metagenomics. A, B, C, D, E, F. It is arranged in such a way. Let's see what is its arrangement. In metagenomics, the genetic material, that is DNA in C. When you say C, this is the DNA. So the genetic material are extracted directly from samples taken from the environment. For example, soil, seed, water, human gut, that is in A. So it can be taken from the soil, seed, or human gut. After filtering in B, it will be filtered in B here. Where is B? This one. This is filtering. Then after filtering in B and sequenced at E, this is a sequencing mission. So by sequencing here, then after multiplication by cloning means making many copies, the same copy. When you copy something, then that will be exactly the same. That in the genetics we call cloning. So by multiplication by cloning in D, this. So these are copied. They are the same. So they are cloned. Cloning by sequencing. These short sequences can then be put together again using assembly methods. In F, here by assembly method, they will be come together. Okay. To deduce the individual genomes or parts of genomes that constitute the original environmental sample. So by this method, we can know which kind of organism is that found in that environment. What is plasmid? This is a circular extra chromosomal DNA molecules in bacteria. Generally, bacterial DNA are circular, therefore, this one is circular, but it is what? Extra chromosomal of the bacteria that can independently reproduce. Plasmids can be used as vectors in recombinant DNA research. Very important area. This plasmid is very important, and scientists use this for research purposes. And they can contain genes important to bacterial virulence. Okay, that virulence means causing disease or pathogenicity. Therefore, plasmid may contain what? The virulence. Or, so bacteria may cause disease because of having this plasmid. So it will be resistant to antibiotic. This is bacteria. This is rod shape, which is bacteria. So this one is the bacterial DNA, the major DNA of bacteria, which is double-stranded and circular. And when you say these are the plasmids, okay, this is extra chromosome. This is the major chromosome. The extra of this chromosome we call plasmids. What is restriction? What are restriction enzymes? These are scissors. Simply consider these as scissors. Proteins that recognize specific short nucleotide sequences in DNA and catalyze cutting at those sites. So, what is the function of scissors? Cutting. So, restriction enzymes are important for cutting the DNA. This is the recognition sites. So, the at promoter region, then there will be transcribed region here. Then there will be RNA polymerase and then single strand template DNA. Then here is the restriction enzyme, like scissors. This is like scissors. This is a DNA. It is not actually simple to cut by scissors DNA. Just this is a simulation. So this is an enzyme, kind of restriction enzyme, E. coli. There is a rule or protocol to name the enzyme. Just let's leave that for a moment. And E. coli is a kind of restriction enzyme. Then it will cut the DNA double strand by recognizing the specific site. This site, for example, it will cut at G and A of this kind of sequence. The sequence should be CTTAAG, and as this site CTTAAG, it will cut the sequence between the nucleotide A and G. Then finally, the product will be this kind. So here is the sticky end, or the end which will be suitable to attach with other DNA base by complementarity, and then there will be multiplication of the DNA. What is clone? This is a group of cells or molecules that are identical by having arisen from a single ancestral cell or molecule. Okay? Because of cloning means copying. So then the result will be identical cell or identical organism or identical molecule. Cloned gene, related with this definition, a recombinant DNA molecule with a gene of interest. Library, just like library, normally we know library, where books are stored. This is also the library where the DNA will be stored. Therefore, a complete set of clones, the copies of the genetic material that contains all the genetic material from an organism, tissue, or specific cell type at a specific stage of development. We call this library. This is an example of clone. For example, this is a sheep, and this one is the other sheep. These are the two kinds of sheep. So, two kinds of sheep, and inoculation, removal of the important part from this mother, and then taking the cell from this one, and then inserting into this mother. We call this surrogate mother. Then there will be production of this kind of sheep. This is a dolly, which is the first animal made by biotechnology or this process. So it is possible to do that. Anyways, clone means copy. Copy of what? DNA. Not a piece of book or a piece of paper or anything, but it is what? A copy of DNA. What is consanguinity? This is having a common ancestor, okay, being blood relatives. Mating between two first cousins, for example, can be termed a consanguineous mating and is indicated in the pedigree by a double bar between the two parents. Such mating can result in an increased frequency of offspring who are homozygous for a recessive autosomal trait possessed by both parents. For example, cystic fibrosis or the amorphic type of RHA nail may caused by this kind of mating. I really hate such kind of mating. Please don't do that. Because when homozygosity increases, then the disease condition will increase. Okay? So that is not advisable by culture or also from this scientific point of view. So don't do that. For example, autosomal recessive disease, let's consider most of the disease in human beings are recessive, so autosomal recessive. When the two recessive alleles come together, then the disease will occur. Otherwise, it will be masked or dominated by the dominant alleles. That is very important, one of the important design of nature. Here is a carrier father. This having white and the blue color indicates carrier. This is a carrier father having this chromosome. This is a chromosome. Then there are genes here, this gene and this gene. And white indicates affected, and the blue indicates unaffected. Therefore, this is here is white and blue carrier, white and blue mother carrier. Then from this couple there will be an affected son or daughter, blue blue, carrier son or daughter, we having white blue, white blue, white blue, white blue. Then affected son or daughter, all white, that indicates the affected one. So the percentage will be 25% probability of the unaffected, 50% probability of carrier, and 25% probability of uh, the affected daughters and or sons. So thank you so much. This is all about my preparation for genetics terminologies. This is not mean that these are only the terminologies in the genetics, but these are very important and relevant. If you can understand all these, which I did in the series of videos, then you will easily understand when you read any kind of genetics books. Thank you for watching. Bye bye.